Good morning, my friends at ANA. I had looked forward to seeing you in person in Chicago until July 21st of this year, when I was riding my bicycle and had a head-on collision with a speeding SUV. Miraculously, my brain, my spine, all vital organs were undamaged, but my lower half was a mess. I have been blessed by the best of modern medicine, which is trauma medicine, and today I'm recording this from my home, still in the early stages of rehab, but looking forward to full recovery. The whole idea of evidence-based medicine has a built-in bias that has distorted health priorities and made Western allopathic medicine what it is. Of course, profitability has driven a great deal of medical practice in the wrong direction, but I'm not talking about the influence of money. The bias I'm talking about comes from the fact that some evidence is a lot easier to gather than other evidence. Researchers in our field face a challenge with validation. Most drug makers target a clinical symptom, and they can determine in a trial lasting just a few weeks or months whether their intervention is working. But we aim to create healthy lifestyles that support a robust, disease-free body over many years. We are optimizing long-term health. Maybe practitioners feel better and function better with good nutrition, but there may be no obvious short-term benefits to some aspects of our program. We're committed to evidence-based medicine, but our evidence may take a decade or more to accumulate. We're limited to observational studies. We can't do experiments because we can't ask a group of people to eat a certain diet for 2 decades on end. We certainly can't ask a control group to not eat a healthy diet for the next 2 decades. There are very few government-funded, comprehensive long-term observational studies from which data can be extracted. They have to guess in advance what data to collect, and if they guess wrong, it can distort health advice for generations. For example, 50 years ago, none of the large studies thought to survey consumption of trans fats, and it required many decades to establish enough evidence to deter the food industry from the convenience of stabilizing foods with hydrogenated vegetable oils. It's far quicker and more convenient to rely on surrogate markers of long-term health than to actually wait and assess long-term outcomes. But the danger is that we make mistakes and choose the wrong surrogates, and then we're drawn into decades of bad medical advice. Here are 4 examples of sound surrogate markers: CRP, C-reactive protein; IL-6, these are markers of inflammation; gait speed, forced exhalation volume for the lungs; HDL, cholesterol. And here are 2 examples of bad surrogates, in my opinion: blood pressure and LDL cholesterol. Large databases from which long-term health factors are studied include the Framingham Heart Study, started in 1948, continuing now in its 3rd-generation cohort. 3,000 papers published to date from Framingham data help to establish that smoking kills. But Framingham was also responsible in part for a flawed theory about cholesterol and heart disease that, again, is dying very slowly, despite accumulating evidence that it is not the right way to think about heart disease. NHANES, the National Health and Nutrition Examination Survey, ongoing since 1971, but with limited information, mostly diagnostics like blood pressure with very limited diet and exercise information, despite the program's title. This has led to major distortions like the decades in which doctors were telling patients to cut back salt to lower their blood pressure. This was revealed 8 years ago to be dead wrong, but still, most doctors persist with the bad advice. The full NHANES questionnaire has thousands of topics, but the only question about diet is, in general, how healthy is your overall diet? The Carefully study is a similar British project. The China study and China study 2 have the advantage of relatively stationary, genetically and culturally homogeneous populations. These have provided a counterpoint to Western diets. The Nurses' Health Study 7 countries study. If you believe, as I do, in tailor-made individual therapies, then it's upsetting to know how little understanding and respect they have in the evidence-based allopathic community. It's not impossible to assess the efficacy of a particular modality or a system of individual diagnosis and treatment, but you have to approach validation with a different mindset. Placebo-controlled, double-blind is not possible, but comparing patients who have been treated with, say, traditional Chinese medicine to patients with similar symptoms treated by Western medicine is very possible, and some of this has been done. Dale Bredesen has been working for decades on an individualized treatment protocol for Alzheimer's disease. RECODE is a system of diagnosis involving genetic, dietary, social, and environmental factors, together with an individual program to address the underlying causes of cognitive decline in each particular case. Bredesen tells how he proposed clinical trials of the program in its entirety and met rigid resistance from IRBs that lacked the imagination to realize it's perfectly possible to test the effectiveness of a system that offers a different treatment to each individual patient. I have tried to set the stage by saying it's difficult but not impossible to assess the long-term effects of the healthy dietary and other lifestyle measures that we believe in. In this context, consider the unique case of interventions that affect the rate of aging, together with new tests that accurately assess a person's biological age. Aging is the single most important factor affecting long-term health prospects. It has been brushed aside in the past only because everyone regarded it as a fixed background about which nothing could be done. Well, now this is starting to change. Measurement of aging is also undergoing a revolution. Methylation clocks offer an opportunity to assess long-term benefits with a trial that lasts just a few months or a year. So, what is aging? The old view of aging is that the body breaks down over time because damage accumulates inevitably. It's physically necessary. The job of anti-aging medicine then is to catalog and prioritize the kinds of damage and engineer strategies for repairing it. We know this view is wrong in many ways, and I've written extensively refuting this view. Briefly, there are many plants and some animals that don't age at all, or even age in reverse, so accumulation of damage is far from inevitable. Furthermore, aging is slower when animals are stressed or starved. This can only be true if they're holding back some of the ability to avoid damage when they are not stressed. In a few minutes, you'll hear more reasons to prefer the alternative view. The correct view is that aging is a programmed stage in our lives, akin to childhood, growth, and puberty. When we're toddlers, growth hormones surge, driving development and growth. Sexual maturity is triggered by a surge in sex hormones. Later in life, menopause and andropause are driven similarly by changes in sex hormone levels. Well, inflammatory cytokines rise gradually with age and gradually destroy the body with inflammation. Apoptosis is dialed up so that we lose useful cells to cell suicide. Meanwhile, all the protective functions of DNA repair and neutralization of ROS are dialed down in age, allowing damage to accumulate that was not accumulating when we are young. Aging is driven by changes in genes, otherwise known as epigenetics. We have the same DNA in every cell of our body, and it doesn't change over a lifetime. But different genes are turned on and off to differentiate one type of tissue or organ from another, and gene expression also changes at different stages of life. This is how the changing hormones, enzymes, and cytokines are orchestrated. I can't say that everyone agrees on this explanation for the cause of aging, but it has been my belief for 25 years, and more recently it has become an emerging consensus among some researchers. Aging clocks. Different people age at different rates, so biological age can be older or younger than chronological age. An aging clock is any system for calculating biological age. There are many markers more or less tightly correlated with biological age, But when we understand that aging is, at its core, an epigenetic process, then we're inclined to trust an epigenetic clock more than any other. How can we measure epigenetics? Well, epigenetics, or gene expression, is controlled in more than 100 ways by the structure of the chromosomes and by molecular tags that are bound to the DNA. DNA is wound around protein spools called histones, and the histones also can have tags that tend to turn a gene on or off. It's all very complicated, and we're just at an early stage trying to understand it. One epigenetic mechanism that we do understand better than others is DNA methylation. In the four-letter alphabet of DNA, A, C, T, and G, cytosine is the C. Cytosine can exist in two forms, with or without an extra methyl group. Methylated cytosine tends to turn off an adjacent gene. It's a signal that says, do not transcribe. Methylation patterns on DNA can be reprogrammed as needed. This can happen in a matter of minutes, or sometimes the patterns can persist for many years, even across generations. Yes, methylation patterns are copied when DNA is copied. So daughter cells can inherit the mother cell's epigenetics. We know so much about methylation because of the accident that there is a simple lab test that lets us map methylation quickly and cheaply. Methylation maps of your DNA are now available from a number of commercial labs for a few $100s. Methylation clocks. In 2013, Steve Horvath, a biostatistician from UCLA, published a landmark paper with an algorithm or mathematical instructions for how to calculate biological age from methylation patterns on DNA. He derived his methylation clock using a combination of statistics, artificial intelligence, and intuition based on a sample of DNA from 7,844 people ranging in age from pre-birth fetal cells to over 100 years. Out of 21,000 methylation sites called CPGs, the procedure identified a subset of 353 that were related to age and found a way to combine them to predict a biological age. The clock worked across a full range of human ages and tissues. The same 353 CPGs that change methylation in a baby are still changing in the same direction in a 90-year-old man. The example of the Horvath clock lent credence to the paradigm that I mentioned a few minutes ago, that growth, development, and aging are part of the same continuum, and the entire process is driven by changes in gene expression that come in part from methylation changes. The computed biological age agreed with chronological age of people in the sample with a median error of 3.6 years. This accuracy is comparable to how well a human might do when meeting a new person and asked to guess his age. And that 3.6 years isn't necessarily an error. Biological age is not the same as chronological age. Some of that 3.6-year disparity represented a true report about who is aging faster and who is aging slower than the average rate. In fact, it was later determined that the Horvath clock, based on methylation patterns, is a better predictor of health and longevity than the chronological age of the same person. I ask you to think about this fact. I find it very significant that an algorithm based on methylation contains more information about health and longevity than the chronological age, which is the only information it had when it was derived. I interpret this to mean that methylation is closer to the source of biological age than simple chronological age is. Chronological age measures the passage of time. Methylation measures gene expression. Again, this supports the paradigm that aging, like growth and development, is driven by changes in gene expression. Horvath has not been idle the last 8 years. He and his colleagues have derived another generation of methylation clocks specialized to different purposes. Some are calibrated to saliva samples, some blood, some for other body tissues. PhenoAge by Morgan Levine and Horvath is calibrated to markers such as inflammation and mitochondrial activity, not just chronological age. GrimAge by Ake Liu and Horvath is calibrated with old stored blood samples, so it is known how long those people lived subsequently, and that's taken as a critical input. There are other specialized methylation clocks for other animal species, and there's a clock that works on 128 different mammal species using homologous CPG sites. Methylation clocks are a work in progress, and they are presently the best measure of biological age that we have. I say this both because they are the most accurate and also because they are derived from methylation, which is one of the core drivers of biological aging. I want to briefly describe 3 studies that have found interventions that reduce biological age as measured by methylation clocks. Then I will end with my own study, just beginning, which will survey the effects of various diets and supplements and exercise patterns on methylation age. First is the TRIM study. I've known Greg Fay for many years, and he's always been interested in decline of the immune system as a primary driver of aging. The thymus gland in the base of the throat is where white blood cells learn to differentiate self from alien, so it can spare the former and attack the latter. The T in T-cell immunity comes from thymus. The thymus gland reaches its maximum size before puberty and then gradually shrinks over a lifetime. Thymic involution contributes to autoimmunity in old age and also to the opposite, decline in the immune system's ability to fight infection. In 1997, when he was 46 years old, Greg experimented with himself as subject using HGH to regrow his thymus. He documented the experience and wrote it up for the Journal of Anti-Aging Medicine. 20 years later, he obtained funding for a clinical trial. There were just 9 men, aged 51 to 65. For 1 full year, they were treated individually with extensive monitoring of many aspects of health and blood factors. In addition to growth hormone, the protocol included metformin, DHEA, vitamin D, and zinc. Part of the rationale for this was that growth hormone can lead to diabetes, and the other supplements were intended to reduce the risk of diabetes. Horvath himself did the methylation clock analysis and was senior author of a 2019 paper in Aging Cell. 4 different methylation clock algorithms showed age reversal. Depending on which clock you looked at, biological age among the 9 men was set back 2 to 6 years. The data was tight enough that all 4 clocks showed good statistical significance, even though there were only 9 subjects. The results are more impressive in that these men were particularly healthy, and they started out with biological age much younger than their chronological years. Their biological age was further reduced from there. The TRIM study attracted attention both among academics and biohackers, and Greg is now well advanced in a follow-on study with dozens of participants and a control group using the 4 supplements but omitting the growth hormone. As before, there is extensive monitoring of individuals with adjustments to dosage based on interim results. Kara Fitzgerald is a naturopathic physician and one of the first to realize the importance of epigenetics for human health. She designed a program to address methylation directly, not with drugs or supplements, but with a diet rich in methyl donors and methyl adaptogens. In addition to the diet, there was an exercise protocol, mindfulness, meditation, and personal coaching. MDL stands for Methylation Diet and Lifestyle. Dr. Fitzgerald's reason for avoiding drugs and supplements was that she was aware that methylation is delicately regulated. The temptation is to supplement with SAMe or B vitamins that are methyl donors, but there's a danger then of hypermethylation, and when tumor suppressor genes are hypermethylated, it can increase risk of cancer. So she chose the path of safety for her first study. The study was conducted with rolling participation from spring 2018 to summer 2019. There were 18 subjects, 20 controls, and I was fortunate to make Dr. Fitzgerald's acquaintance just as the data were coming in, and I did the data analysis for the study myself. According to the original 2013 Horvath clock, subjects were an average 2 years younger after just 8 weeks in this program. Other clock algorithms did not reveal benefit. There was an indication that subjects who had one or more copies of the MTHFR SNP may have benefited especially from the program. MDL results were less dramatic than TRIM qualitatively, but there are also major advantages of MDL. It's cheap or free, it's safe, and results accrue in a very short time. Dr. Fitzgerald is conducting a large follow-up study guiding subjects through her clinical practice with the help of a cell phone app developed explicitly for MDL. Blood samples will be used to evaluate methylation age because calculations are more accurate than for the saliva or buccal samples used before. Dr. Fitzgerald's book, The Younger You, is due out in January. Harold Katcher's plasma fraction. Could it be that a scientist with meager lab resources, but who deeply understands what aging is, can leapfrog ahead of government and tech startup companies with aging labs that have billions in funding? Well, Harold Katcher is semi-retired from a successful but uncelebrated career as a PhD biochemist. He was one of a team of scientists to discover the BRCA gene in 1994. Katcher became intrigued with parabiosis studies about 15 years ago and wrote a paper about use of blood transfusions for rejuvenation. In parabiosis, two mice are surgically joined so they share a single circulatory system. These bizarre experiments have been performed for 150 years, but they only reached the scientific mainstream after they came from the Stanford laboratory of Tom Rando around 2006. If a young mouse and an old mouse are joined in this way, tissues of the old mouse show signs of rejuvenation, while tissues of the young mouse evince premature aging. This raises the possibility that signal molecules in blood plasma are transmitting information about the age of the body, and cellular aging is a response to central signaling. This represents a paradigm shift from those who thought the root cause of aging is damage that accrues at the cellular level. The new paradigm is that aging is a systemic function, centrally controlled. Tissues of the body respond flexibly to signal molecules in the blood. The tissues can be rejuvenated or they can be aged, depending on combinations of signals that they receive. The laboratories of Irina and Mike Comboy at Berkeley and Amy Wagers at Harvard have become closely associated with this idea. Work continues at Stanford by Rando and by Tony Weiss-Correa. Katcher had an idea what the particular signal molecules might be that centrally dictate the age state of the body. In 1996, he obtained a very small investment from an Indian entrepreneur and moved to Mumbai for a year, where experiments could be performed at reduced cost and without regulatory interference. His very first round of experiments was strikingly successful in transforming the physiology and behavior of 2-year-old rats. Katcher has reported increasing success with his rejuvenation experiments ever since. Last year, he published a preprint in which Horvath was the senior author. The methylation age of his 2-year-old rats had been transformed to match that of 1-year-old rats. Many markers of metabolic age, including inflammation, mitochondrial activity, strength, and learning ability, simultaneously showed signs of rejuvenation. In the latest trial, old rats were made young, then housed for 6 months until they got old again, and they were rejuvenated successfully a second time. Katcher has teamed up with Greg Fay, author of the TRIM trial above, to test his rejuvenation protocol in dogs, recruited from people with aging pets. This trial is underway right now. Horvath and his postdoc, Ake Lu, have designed a methylation clock especially for dogs to make this possible. So, what is Harold Katcher's secret sauce? He will say only that it involves a few intravenous infusions spaced over 2 weeks. It derives from fractionating blood plasma, and he says it is easily synthesized and should not be expensive in bulk once manufacturing facilities have been set up. Secrecy and intellectual property concerns have prevented the collaboration of large numbers of laboratories that would certainly be interested in replicating and extending Katcher's results. Their expertise has been locked out, at least for now. Katcher's company is called UVAN, and his plasma fraction is called E5. His book, The Illusion of Knowledge, was published earlier this month. Katcher and his corporate director are planning to try E5 with their first human volunteers within the next few months. DataBETA is my project about to recruit our first subjects this fall. The acronym means Database for Epigenetic Evaluation of Treatments for Aging. Hundreds of thousands of anti-aging enthusiasts and biohackers around the world are trying diets and supplements, drugs and fasting programs to try to stay younger longer. These people constitute a natural experiment if only we can measure their success. Well, methylation clocks provide the means to do just this. Our plan is to recruit 5,000 people, aged 45 to 65, who are doing interesting things with the intent of extending their lifespans. We will aim for a diverse array of strategies, and in particular different combinations of strategies. It's my personal conviction that if there were a single silver bullet that stopped or reversed aging, we would have found it by now. More likely, there are combinations of drugs, diets, and other programs that synergize together to create a substantial effect. Therefore, we've developed an extensive questionnaire that delves into every aspect of a subject's life that's possibly relevant: eating, sleeping, work, social life, exercise, psychological counseling, meditation, yoga. All our subjects will undergo a methylation test at the beginning and then the end of a 2-year period. We expect a wide range of effectiveness. Perhaps the average will be 0. It's very possible that most of what people are doing doesn't work at all. It may be that for a lot of our subjects at the end of 2 years, the methylation test just shows they're 2 years older. However, we hope that there will be some combinations of treatments that have a large effect. And at the tail end of the distribution will be people who are epigenetically younger at the end of 2 years than they were at the beginning. Our analysis will focus on these people, the most successful 5%. What strategies do they have in common that distinguish them from the other 95%? If this succeeds, we should have one or more candidate strategies for rejuvenation, which can be further tested with follow-on studies that look more like a traditional placebo-controlled clinical trial. DataBETA has received generous seed funding from Didier Colonel and the Hielis Foundation. We anticipate crowdfunding for most of the $3 million budget. This is an open source project, and all of our anonymized data will be shared freely so that other researchers can mine the data for things we have not found. Here's what I hope you take home from this presentation. It's challenging to validate practices and interventions that are intended to optimize long-term health. There's a new opportunity to do this by focusing on biological age. This is possible because, first, biological age is a powerful, in fact, exponential predictor of the most important chronic diseases. And second, Biological age is now measurable with new technology based on DNA methylation. Methylation age tests have already been used to validate 3 anti-aging interventions, 2 in humans, 1 in rats. I'm launching this fall a broad survey of common and not so common anti-aging practices using methylation age to assess whether any of them are effective and to search for combinations of anti-aging practices that synergize. This concludes my presentation. Thank you so much.